because both the speakers were my university batchmates and very good friends. Still now, we are very well connected. So without much delay, I would like to call upon our chief patron and principal, sir, Dr. Shomen Chando, to welcome the speakers and delegates. Over to you, sir.
Thank you, Sanjukta. Am I audible?
Yes, you are.
Okay. A very good afternoon.
শোনা যাচ্ছে না. I think we can't hear you.
Today's webinar titled Biology, Behavior and has been arranged by the Department of Zoology in collaboration with the IQAC of our college. Thanks to the organizers. As far as my understanding goes, today's two lectures are of different kind. Dr. Sanjukta, please correct me if I'm wrong. Although I am not a person with a suitable background, I hope you will allow me to say a few words. Please bear with me. Recent trends in research on interspecific interactions have unveiled several important aspects regarding the interplay of the component species, investigating the ecoethology of one component species and its trajectories can provide adequate information on the other. Human-animal interaction is one such field that attracts researchers to find solutions for evolving problems like human-animal conflict, spread of zoonoses, uncontrolled population growth of unwanted species, etc. In the recent times, studies on human-animal interactions have enabled us to grasp evolutionary processes like domestication. Such scientific investigations, coupled with of species. As a first domesticated species, interacting with humans. Thus, exploring the dog-human interaction paradigm is especially helpful to analyze the underlying dynamics of the domestication process that enabled the transition of wolf-like ancestors to man's best friend. Domestic dogs, Canis lupus familiaris, are remarkably sensitive and responsive while interacting with humans. Pet dogs are known to have social skills and abilities to display situation-specific responses. But there is lack of information regarding free-ranging dogs, which constitute majority of the world's dog population. Free-ranging dogs found in most of the developing countries interact constantly with familiar and unfamiliar humans, receiving both positive and negative behavior. Thus, understanding human intentions and subsequent behavioral adjustments are crucial for dogs that share habitats with humans. Here, we subjected free-ranging dogs to different human social communicative cues, friendly and threatening, low and high, followed by a food provisioning phase, and tested their responsiveness. Dogs exhibited higher proximity seeking behavior as a reaction to friendly gesture, whereas they were prompted to maintain distance depending on the impact of the threatening cues. Interestingly, only the high impact threatening had a persistent effect, which also remained during the subsequent food provisioning phase. An elevated approach in the food provisioning phase elicited the dependency of free-ranging dogs on humans for sustenance. Our findings suggest that free-ranging dogs demonstrate behavioral plasticity in interacting with humans. Now, on a slightly different note, Daniel Cousins quoted in a BBC article titled "Humans Aren't Alone in Farming Crops and Meat" that these creatures have mastered agriculture, cultivating crops, and herding livestock. In the process, all parties involved have reciprocally shaped each other's evolution. For humans, farming changed everything about 10,000 years ago. The domestication of plants and animals meant people could, for the first time, build up food surpluses. This liberated them to think about something other than subsistence. Nomadic hunter-gatherer lifestyles gave way to the permanent settlements. Villages became cities, gave rise to civilizations. If we look closely enough and see we are not the only animals that can grow our own food. In fact, ants and beetles were cultivating bumper crops of fungi 50 billion years before we started farming. And just as we invented agriculture several times in different parts of the world, when we made use of plants and animals living wild around us, several non-human species have developed farming-like relationships with organisms they have encountered. Some cultivate crops, others have mastered animal husbandry. Either way, all such relationships are examples of a rare form of symbiosis known as a cultivation mutualism. In many ways, these associations resemble human agriculture, although there are limitations to the analogy. These days, we consciously carry out sophisticated selective breeding programs to maximize crop yields or improve livestock. That's not true of animal farmers, whose associations have been forged unwittingly by natural selection. We also raise multiple crops, whereas animals typically specialize in one. Nevertheless, in the co-evolution of organisms in farming-like mutualisms, there are similarities to the way we first domesticated plants and animals in the Neolithic. We can also see parallels in how such associations can change the biology of both parties. And as with humans, agricultural life has, in some cases, allowed animal farmers to rise to major ecological importance. Although it's not at all a well understood subject, for me at least, we all are eagerly waiting to hear the stories from our outstanding speakers in this evening. With these few words, I welcome you all again and announce the session open. Thank you.
Thank you, sir. That was indeed a very warm welcome and a very interesting one. Before we go to our technical session, I would like to request Dr. Programay Pal, Coordinator, IQAC of Fakir College, to say a few words. Sir, please.
Thank you, Sanjukta, for your kind introduction. A very good evening to all. At first, let me give the opportunity to welcome you all to this webinar on behalf of our Fakir College. As you all know, today's webinar has been organized by our Department of Zoology in collaboration with the IQAC of the college. We are really honored to get our two distinguished guests, Dr. RUCHIRA and Dr. ANINDITA with us. Fakir College family cordially welcomes both of you, madam. Our principal, sir, has nicely briefed about today's subject in his inaugural speech. I am also not a person of this field at all. What I have been able to grasp in my little knowledge is that animal behavior is the bridge between the molecular and physiological aspects of biology and the ecology. Behavior is the link between organisms and environment and between the nervous system and the ecosystem. It is one of the most important properties of animal life. It plays a critical role in biological adaptations. Behavior is how we humans define our own lives. It is that part of an organism by which it interacts with its environment. The beauty of an animal includes its behavioral attributes. Many problems in human society are often related to the interaction of environment and behavior or genetics and behavior. With these few words, I think I should stop here, as I know that all of you are eagerly waiting to hear from our honored guests who are experts in this field. Once again, I am welcoming all of you. Thank you very much.
Thank you, Dr. Pal. Well, our inaugural session ends here. Now we move towards our technical sessions. Each talk will be followed by an interactive session for which you may use the chat box to type out your queries. Kindly turn off your audio and video for facilitating smooth transmission of the webinar. Now I will hand over the baton to my departmental colleague, Mr. Bappaditto Patro, for the first session. Bappaditto, over to you.
Thank you, ma'am. Good evening, all. Myself, Bappaditto Patro, welcome you all in technical session for today. In this ornamental day, I am blessed to get the opportunity to introduce our distinguished speaker, Dr. RUCHIRA Madam. Dr. RUCHIRA Madam was graduated student from Lady Brabourne College and completed her post-graduation in Zoology from University of Calcutta. She completed her PhD in Behavioral Science from IISER Bangalore under the guidance Professor Raghavendra Gadekar. Dr. Sen pursued postdoctoral research at University of Purdue as well as University of Texas before joining as an assistant professor in Guru Gobind Singh College, Chandigarh. Her current research focuses on behavior and molecular biology of paper wasp and fungus growing termite. She has over 20 publications on various aspects of termite and wasp and has her own YouTube channel focusing on educational videos of the same. Dr. RUCHIRA Madam was also a guest lecturer in Fakir College, Zoology Department for some time. So it is indeed a very special moment to have her amongst us. Today she will enlighten us with her talk on agriculture in non-human societies. So it is going to be a very interesting talk and all we are eagerly waiting for this. Ma'am, please.
Thank you,
Thank you,
Doctor,
Doctor,
for
for
this
this
kind introduction. Honorable Principal, Honorable IQAC Coordinator, eminent teachers, all my dear students present here, and my two very good friends, Shangjukta and Anindita. It is a great pleasure for me to give a talk here today, especially because, as Dr. Patra said, that I was a teacher at Fakir College. So this was my first job. Between my MSc and PhD, I worked there for about 5 months. And I still remember the train journey from Kolkata to Diamond Harbour and all the teachers there, all the students there, and especially the college, very fondly and very frequently. So although it is just a virtual presence amongst you, it is a huge honor and pleasure for me. So without further ado, let me present my screen and let's talk about agriculture in non-human societies. Okay, so let's first start with what is agriculture. Agriculture is basically production of food by growing crops or by raising livestock. Now, we humans invented agriculture about 10 to 12,000 years ago, right? And this invention has allowed us to shift from hunting and gathering from nature to manipulate nature to produce our own food as well as to get a steady supply of food. And this, as your principal, Dr. Chanda mentioned, that this has been proposed to be one of the major reasons for the settlement of civilizations of humans. So we understand the importance of agriculture in human societies. But today we are not going to talk about human societies. We are going to talk about non-human societies. And when we talk about non-human societies, those of you who are not from biology background, first thing that might come to you is a group of chimpanzees or a herd of elephants or a herd of deers or something like that. But in technical biological terms, we do not really call them social. We call them group living organisms, but they are not truly social. There are some organisms, however, whom we call truly social. And those organisms include the ants, termites, some bees, and some wasps. These species or these groups of insects, they belong to the group of truly social organisms. Now, agriculture is seen in many of these societies. Not all of these insects practice agriculture, of course, but there are some ants, some termites, and some beetles who have invented agriculture. Now, I haven't shown you a picture of beetles here in this slide, and the reason is the Ambrosia beetles, or the group of beetles which practice agriculture, they are also not included among truly social insects. And this slide is showing all the true insect societies. But there are, again, some beetles which practice agriculture, and I'll also talk a little bit about them a little later. Now, as I said, that some organisms, some social insects practice agriculture, and that includes some ants and some termites. These ants and termites have invented agriculture about 50 to 60 million years ago. We humans invented it about 10 to 12,000 years ago, but they have invented it 50 to 60 million years ago. So they have invented it much earlier than we have invented it. Now, second thing is that although we have multiple and variety of crops, including the cereals, pulses, vegetables, fruits, etc., the insects do not have that much variety. Insects actually form fungus. So they have certain types of fungus with whom they have formed a symbiotic relationship, and they form a garden for these fungi, and they cultivate fungi in these gardens. So where do they get this fungus? When the new queens leave the colony, they carry some fungus with them. In case of ants, they carry the fungus in their mouth, and in case of termites, they carry the fungus in their gut, that is in their digestive system. So when they start their own colony, they use that fungus as the seed and they start their own garden. Now, here in this picture, in the left side, you can see the tufts of white cottony stuff, and that is the fungus that the ants cultivate. And here in this side, you can see these small ball-like structures. This is the fungus that the termites cultivate. Now, both of these belong to Basidiomycete fungus, and they have certain similarities and many dissimilarities too. Now, as I said, that I'm also going to show you the beetles. Just a second. Okay, so one more thing here, that here in this picture, you can see small green parts. What are these green parts? These are leafcutter ants, and they cut small pieces of leaves and they use that leaf as the substrate for the garden. So as we add manure, as we add soil for cultivation, they also collect vegetative material on which they cultivate fungus. So here you can see a picture of leafcutter ants who have cut small pieces of leaves, and they carry that as flags in their head, and they use that to form the garden. And in that garden, they cultivate fungus. Now, here is a picture of a species of Ambrosia beetle. And these Ambrosia beetles, they form galleries inside wood, and they cultivate fungus in there. Their fungus is of Ascomycotina group, and this fungus is a little different from the fungus that the ants and termites cultivate. But nevertheless, all these insects use this fungus for their food. Okay. Now, where are these amazing insects found? The beetles, they are cosmopolitan. They are found more or less everywhere. But the termites and the ants, they have very specific locations. The ants are found in the New World, that is in American continents. They are particularly found in southern part of North America and northern part of South America. And the termites are found in Africa and Southern Asia. So what does this tell you? This distribution, that the ants, fungus-growing ants are found here and the fungus-growing termites are found here, this basically tells you that they have evolved separately. They have invented agriculture separately and independently. See, remember one thing, ants are found everywhere and termites are also found everywhere. However, the fungus-growing termites are found only in this part and fungus-growing ants are found only in this part. Australia, thankfully, is devoid of any of these insects, but they have some beetles which cultivate fungus. And why I say that they are lucky is because these are all considered pests. Because you saw that fungus-growing ants, some of them cut leaves, and they can chop off leaves from very huge trees in no time. So they are also considered as pests. And those of you who have studied about wheat pests, you must have studied Odontotermis obesus, because they also damage the roots of wheat and roots of many other trees, because they take those vegetative materials to use as substrate for their garden. Okay. Now let's go back to human agriculture. Human agriculture has many steps involved in it. to turn these green crops into these golden crops, we need to go through a huge number of steps. First step is, of course, acquiring the seeds and choosing the right type of seed, right? And then you also have to prepare the soil to cultivate the seeds. And then you have to sow the seeds. Then you have to add manure and fertilizer so that your plants get enough nutrients. Then you also have to remove the weeds and pests so that they do not damage your agricultural fields. You have to arrange for irrigation. You have to arrange for harvesting the crop. Now, if we are comparing insect agriculture with human agriculture, then there should be at least some of these steps available in insect agriculture as well. And it turns out that most of these steps are seen in insect agriculture as well. Now, I personally haven't worked on all of these aspects, but others have. I have particularly worked on choice of seeds or choice of cultivar in insect agriculture and the aspect of removal of weeds and pests. Okay, so we will talk about that. First, we are going to talk about choice of cultivar. So to understand why the choice of cultivar is important, let's look at the insect agriculture system a little bit in more detail. Okay, so these insect farmers, be it ant, be it termite, be it beetle, anything, they cultivate the fungus and they get their food from the fungus. What is the fungus getting in return? Why should they become the food of these insect farmers? The fungus are getting nutrition, shelter, and disease protection from the insect farmers. Okay, so this is very important for both the partners. Here we will call them the host, the insect farmers, we will call them the hosts, and the fungi as the symbiont. Now, the success of any of this symbiotic association will depend on the choice of the symbiont. Because if the fungus does not produce enough food, then the insects are going to suffer. If the insects are giving all the nutrient, shelter, disease protection to the fungi, they should also expect to get a huge production in return, right? So whether they are choosing the right fungus to cultivate, that is very important. And for my first experiment, I worked on this aspect. So I wanted to see if given a choice, would the ants choose their cultivar or not? Now, why is that question important? Because if you just dig out the ant colony, then you would find many fungal gardens in that colony. So I'll show you a picture first. Leafcutter ants' nests from outside looks like this. You can see that there are soil taken out from inside and they are dumped outside. Okay, so there are small points of entry around this dumps of or the heaps of soil. And if you dig this, like this one here, if you dig a hole, big enough hole, then what you see is there are small chambers inside the soil, okay, or under the soil. And in these small chambers, we find these gardens. These are soft honeycomb-like structures, and there is a lot of fungus in these substrates. Okay, so this is known as a fungal garden. There are millions of ants in these gardens. They are continuously taking care of the garden and they are cultivating their fungus for food. You can see that if there are millions of ants in these gardens, they need the food. Now, ants being the adult insects, they do not eat much, but their brood eat a lot. And their brood are raised solely on fungi. Okay, so they have to produce a lot of fungus. Now, if we collect fungus from any of these chambers under one particular nest, then what we see is that there is a monoclonal strain throughout the garden. What does the monoclonal strain mean? That if you sequence that fungus, if you select a particular gene, and if you sequence that gene, it tells you that it is exactly the same strain throughout the colony. Okay, so if it is rice, then it is exactly same variety of rice throughout. If it is Sona Musuri chawal, then it is Sona Musuri chawal throughout the garden. Okay, so that is what it means. It is genetically similar throughout the colony. Now, if you dig out another colony, you might find another strain, especially if you go to another state of America and if you collect the fungus from that nest, from there, if you collect an ant's nest and if you collect the fungus there, then you might find another strain. And these two strains might be different, but within the same nest, you would find the monoclonal strain or the same strain throughout. Okay. All right. Now, obviously, every time you need to do an experiment, you cannot go and dig a colony, right? So what we do is we go to fields time to time and we dig the colonies and we keep them in lab. Okay, so these are each one separate colony in these boxes, and if I open the boxes, then this is what we see. We try to mimic the ant chambers in the soil with plastic boxes, and these plastic boxes are connected with plastic tubes so that the ants can go from here to here to here to here. Okay, and this we call as the feeding chamber, where we give them leaves, sometimes Quaker oats, sometimes orange rind, etc. So the ants collect their nutrient from here and they form their substrate here and they cultivate their fungus here. So they do not eat any of the material that we give here. This is, although this is called a feeding chamber or a foraging chamber, they do not eat this material. They feed only on the fungus, but they use those to make the garden here. So we have these separate colonies in our lab, and we can collect the fungus from these colonies when we have to do the experiments. Okay. Now, if we open the box, this is the queen and these are the workers. Okay, so this queen is responsible for laying all the eggs in the colony, and the workers are responsible for taking care of all those eggs as well as for their agriculture. Okay, now we have got the hosts, we have the ants, we need the fungi. Okay, so we collected fungus from many different parts of US and Mexico as well. And there, from there, we collected different genotypes of fungi. Okay, so we had in our lab 8 different strains of fungi. So for a choice experiment, we had to give them a choice of fungi, and we wanted to see if the ants would take any particular fungus and will make their garden out of it. Okay, so we had 2 sets of 8 fungi. So we used them as their cultivars for choice, and we use the Atta texana ants for the hosts. Okay. So how do we use this fungus? There was another technique. Why I'm going into the methodology is just to give particularly the students an idea about how you can carry out these kind of experiments. You can test whether the ants would choose something or not. These kind of experiments are not possible to do in nature, but you can do it in your lab. Okay, so that is why I am going into the detailing of the experiment, how you design the experiment. Okay, so we had these 8 different sets of fungi, and each fungus we blended with potato dextrose broth, which is a media, and then we spreaded it on sterilized oats, and we grew the fungus for about 3 weeks. These cultivar fungi, when they are not under the care of ants, they either do not survive, or if we give them proper nutrients, they survive, but they grow really, really slowly. Under the care of the ants, they grow pretty fast, but under our care, they do not grow very fast. Okay, so we had to keep them for 3 weeks. Now, there was another complication. In 2005, a group in Denmark had shown that Acromyrmex ants which is another species of leafcutter ants or another genus of leafcutter ants, they have a mechanism of keeping out any diversity of crops. So what happens in these species is that the species that they worked on is known as Acromyrmex echinator. And what they showed is that when Acromyrmex echinator feeds on their cultivar fungus, the cultivar fungus goes into their stomach, and then when they defecate, they have some incompatibility material in that fecal matter. And when they defecate in the garden, if there is any foreign fungus, that foreign fungus, when it comes across the fungal incompatibility factors from the ant's fecal matter, the new fungus would die. Okay. So that should actually solve all the problems. If you have, if you like a particular type of rice and you eat that rice, and when you defecate in your garden, your fecal matters take care of any other kind of crop. Okay, so any other kind of crop cannot grow if you defecate in the garden. It sounds very gross, but think about the ants. They are eating, cultivating and defecating everything in the same place. And their fecal matter actually works as the manure for the garden. Okay, it is fertilizer. Okay, so it is not as gross for the ants. Anyway, so the point is that this group from Denmark, they showed that they have these incompatibility factors, and that takes care of any other foreign fungus in the garden. Okay. So what we did is that we took the ants and then we tried these ants directly from the garden, as well as we kept some ants for 7 days on sugar solution diet. When they feed on sugar solution, what happens is that they are defecating for those 7 days, right? So they won't have any other incompatibility factor in their fecal matter anymore. So for 7 days, they haven't eaten any fungus. They have only drank sugar solution. And then we take them and we give them a choice of 8 different fungi. This one is known as a Cafeteria Experiment, and in this Cafeteria Experiment, I gave them small pieces of fungi grown by us on oats. Remember, I said that these 8 different genotypes were grown on oats, and those were all sterilized oats. So this is basically a monoculture, okay? Monoculture of 8 different genotypes, and we gave them in this kind of an arena. Okay, so this is basically a plastic Petri plate, and on that we had, mimicking the soil, we had plaster of Paris, and to mark differences between the gardens, we had plastic shields, okay, so that the fungi are not mixing with each other by air or by anything else except by the ants. Okay, now we kept this kind of experiments. We put some ants at the center. We gave some substrate. Here we gave some oats for the ants to add into the garden. And we had these 8 different small pieces of fungi, which the ants were to use if they wanted to make a garden out of it. So what would happen here is that the ants would get a choice of all these 8 different fungi, and they can select whichever they like, and they could make it into a garden. So we were expecting that they would select a particular type of fungus, but we did not see that. What we saw is that they indeed made a garden, but they cleared out many different gardens. Some they did not touch at all. Some they made into a trash pile, and some they incorporated together to make another garden. As a result, when we took fungus from this garden, which was made by the ants, we saw different genotypes put together. So it's like a chimeric garden, okay? So it's basically like a mixture of all genotypes, or not all genotypes, some preferred genotypes, which were put together, and the ants made a chimeric garden. Now, this is very different from what we were seeing in nature, as well as what the Denmark group had shown. Okay. Why is our result so important when we see the chimeric gardens? Firstly, this is different from natural colonies. In natural colonies, you just find one particular strain. But when we are giving them a choice, they just mix it up and they make a chimeric or a mixed garden. Okay. This is particularly significant because if you try to grow two fungi together, there is a chemical warfare between them. So if you try to grow two fungus in a media without the ants in lab, then what happens is that the two fungi would spray different chemicals to each other, and as a result, the place where there is a contact zone, both the fungi would kind of burn and it would become black. Okay, so there is a war between the fungi if we try to grow them. But when the ants were growing them, there was no chemical warfare. These were pretty healthy colonies. Those survived for 2 weeks, 3 weeks, 4 weeks till the ants died. Okay, so the ants have a certain lifespan. After that, the ants would die. And there might have been some bacterial infection in some cases, but the gardens were pretty healthy. We had no problems with that. Okay, so see, the ants have a mechanism to stop any chemical warfare between two fungi. Okay, and there was no effect of fresh ants' feces on foreign fungi, because the Denmark group showed that if they defecate on a foreign fungus, then that foreign fungus would die. But in this case, we tried with purged ants who were maintained on sugar solution for 7 days before, and some ants which were freshly collected from the garden. Both groups made chimeric gardens or mixed gardens. Okay, so there was no problem when they were freshly taken from the garden. They must have defecated on their garden, but that fecal matter did not have any effect on the garden. So our results were quite different. Okay, now we are talking about Symbiont Association, right? So we have talked about the fungal symbionts. Now, the fungal symbionts are not the only symbionts in these agricultural colonies. There are some bacterial symbionts as well. What happens here is that when we have a symbiotic association of ants and fungi, this works for both of them. Ants are giving them nutrition, shelter, disease protection, and fungus is giving them food. Fantastic. But in any nice association, any successful association, there will be some parasites. And fair enough, in this case also, we find many fungal parasites, okay, like Trichoderma, Fusarium, Syncephalastrum. But most important is Escobopsis. If you take a colony of ants, if you take the garden of the ants, and if you remove the ants, then within no time, the garden will be completely infested by Escobopsis fungus. Escobopsis is there all the time, but when the ants are there, there is a control of Escobopsis. But if you remove the ants, then turns out that Escobopsis takes over all the fungus, takes away all the nutrients, and spoils the whole garden. Okay. Now, in a groundbreaking study, Cameron Curie et al. Showed in 1999 that fungus-growing ants have some antibiotic-producing bacteria here. Okay, they showed that there is a white stuff under their prothorax, and if you check this area, if you take this part and if you grow it on a plate, then what happens is you get some bacteria which belongs to genus Pseudonocardia, and Pseudonocardia is an antibiotic secreting bacteria. Okay, now this antibiotic, they showed that it can kill Escobopsis. It does not harm the cultivar fungus, but it destroys the Escobopsis. Okay, so this is an amazing study. It shows that the ants have their own innovation. They have their own pesticides. Ascovopsis is a pest, right? We need to make DDT, BHC, aldrin, this, that, endosulfan, all those things, which then we have to first synthesize, then we have to ban, we have to go through all that process in our agriculture. But these ants, for millions of years, they have their pesticide on their body, okay? So they have these pesticides under their prothorax, which is a culture of bacteria. So they provide nutrition, shelter, and disease protection to this bacteria as well. And these bacteria secrete some antibiotics which can be used to kill Ascovopsis, okay? So they also showed that there is an ant Ascovopsis and Pseudonocardia co-evolution model, and this model was based on 4 key observations. First one was that there is a single lineage of Pseudonocardia bacteria in per colony. Okay, so the queen has it. The queen gives it to all the workers when she first develops a colony, and then when new queens are formed, these new queens take that Pseudonocardia lineage under their prothorax and they start their own colony. Okay, and this goes through vertical transmission. Okay, so they have only one strain of Pseudonocardia and it goes from queen to the new queen and so on. So there is vertical transmission. It is not found in males. So if you say that males are coming from a different colony and they can bring their own Pseudonocardia, that is not what they were suggesting. So another thing they said is that these bacterial secretions actually promote cultivar growth and they inhibit Ascovopsis growth. So the fungus that the ants want to cultivate, these bacterial secretions help them growing and they stop the parasitic fungus. Fantastic model once again. Okay, so we tried to retest these models. And for that, and we wanted to go into more detail of this system, okay? So what we did is that we collected ants from field or from our lab colonies in sterile saline buffer. And this saline buffer basically maintains the pH and you can put the ants in that, the ants die in some time. And then you vortex them. When you vortex them, what happens is whatever is outside the ants, whatever bacteria is outside the ants, they are continuously shaken in this buffer. As a result, the bacteria will go into the buffer. Okay. Then we take that buffer and we spread it in a minimal media plate. See, ants are covered with cuticle. Those of you who are biology students, you know that cuticle is very, very nutrient poor medium. Okay, so what we did was that we made nutrient poor medium. Most bacteria, most fungi would not grow in these plates. However, Pseudonocardia, being adapted to survive on ant skin, they could actually grow in these plates. So we wrapped these plates and kept them for a few weeks. And then what we saw is that the ants would, the colonies of Pseudonocardia bacteria would grow in these plates. Okay. And we, from different ant colonies, we found many different bacteria, not just one type of Pseudonocardia. We actually found Pseudonocardia as well as Amycolatopsis. Okay, and there were many other types of bacteria too. So this is called the culture dependent mechanism of isolating bacteria. We used culture dependent as well as culture independent mechanism. Culture independent mechanism basically takes the ants and then it sequences for a particular gene all the bacteria that are present on that ant. As a result, you would get sequences of millions of bacteria on the ants. And we saw that Pseudonocardia was not the only bacteria that were present on the ants. There were many other antibiotic secreting bacteria present on the ants. And in culture dependent mechanism, we found many different strains of Pseudonocardia and some Amycolatopsis strains as well. Amycolatopsis is another bacteria which produces antibiotics. We also found Streptomyces, which is another bacteria which secretes antibiotics. Okay, so what we saw is that not only workers had the bacteria, the garden had the bacteria, which is understandable. If the ants were using it to kill Ascovopsis, then when we tried to isolate bacteria from the garden, we found them. We also found them from the males. Okay, so here W means workers, M means males, female is for F, and G means gardens. So we found bacteria from all these different things. So when we find it from the males, that means that not only it is going from one queen to the other, but males from a different colony can also bring their Cyrnocardia strain, and they can transfer it to the females while they are mating. Okay. Also, we not only found just one strain. Here you can see that Cyrnocardia, a particular strain, was found in many different colonies, and one particular colony had many different strains of bacteria. Okay, so for each one, we had many different strains, except for this Trachymyrmex zetechi colonies. We had in all the different fungus growing ants, we actually tried different types of fungus growing ants, not only leafcutter ants like Atotexana, but there was Scyphomyrmex wheeleri, Trachymyrmex turifex, Trachymyrmex zetechi, Mycocephalus smithii, Sericomyrmex ambelis, etc. Different things, whatever we had in lab or whatever we could find from the field easily, we took all those things and we tried to see what are the bacteria they have. Okay, and we found different strains of bacteria in the ants. So culture independent mechanism, those of you who know about it, 454 pyrosequencing method was used, and this data, 454 pyrosequencing data also showed that ants had many different types of antibiotic secreting bacteria on them. Okay, so then we wanted to see what is the role of these different bacteria on the fungus. Okay, so for that we did another experiment where we just tricked some bacteria at the center of a Petri plate, kept it for a few days so that the bacteria would grow and it would secrete any antibiotic that it has, and that antibiotic will go along the media. Okay, and then we put 2 plugs of fungi, same type of fungi, in the plate. Okay, and then we saw how the fungus was to grow. In some cases, the fungus did not grow at all. Okay, so we call that no growth. Sometimes the fungi would grow a little bit, then it would stop. Sometimes the fungus would grow so much that it could even touch the bacteria. Sometimes the fungi did not care about the bacteria and it grew all over the plate. So we gave them different names like no growth, attenuated growth or arrested growth, bacteria touch growth, and full growth. Okay, and these plates sometimes even looked like that old Durdarshan model. Okay, so they would grow everywhere except they would avoid this area. Okay. Now what we did is that we tried this for our all the bacteria that we had, so 12 different bacteria, and we tested them against 22 different types of fungi, and these are known as pie charts. So for each bacteria strain that we found from our colonies, we had tested 22, or in some cases a few less, fungi to see under the influence of the bacteria, how are the fungi growing. The red ones, red and the pink, means that there was no growth of the fungus or there was arrested growth of the fungus. The green ones means that the fungus could touch the bacteria or it could fully grow on the plate. Okay, so all the red parts would basically mean that the bacteria had some effect on the fungus, and the green parts would mean that the fungus had no influence of the bacteria or had very little influence of the bacteria. And see, in each of these pie charts, the red part is more than the green part. That means that each of these antibiotic secreting bacteria, it could be Pseudocardia or it could be Amycolatopsis. These two are Amycolatopsis, this one and this one. They had the ways of stopping the fungus. Okay, so in the first paper that Cameron Curie et al. Had published, they had shown that the bacteria was specific for only Ascovopsis. But we showed that it not only kills Ascovopsis, but it also stops the cultivar fungi. It also stops entomopathogenic fungi, which means that the insects get infected by certain fungi. So these bacteria can stop those entomopathogenic fungi too. They can stop garden pathogens. They can also stop endophytes. Okay, so they had a generalized antifungal activity. Okay, they would not only stop Ascovopsis, but they would stop many different types of fungi. Okay, so we showed basically that the workers from the same nest carried different strains of Pseudocardia, not just one. We showed that through culture dependent as well as culture independent surveys. Then we showed that males carry Pseudocardia too. So horizontal transmission is also possible, not just from queen to queen, but males can also add to the new colony. And Pseudocardia is not the only antibiotic secreting bacteria present on Attine ants. They have many other antibiotic secreting bacteria, which may be very close to Pseudocardia, but they are a different genus. Okay, and they have very generalized antifungal activities. Okay, now all these parts were done in Texas, and when we came to India, when we wanted to try it, but obviously there is no fungus growing ant in India, but there are fungus growing termites, right? So there is Odontotermis obesus. This is one termite colony, which you must have seen, these kind of mounds. They are usually called ant hills, but these are nests of termites. Okay, so here also, if you open the garden, then what you see is that these gardens, if you open those mounds, then you see there are galleries, and inside the galleries, there are fungal gardens. Okay, so the termites, just like the ants, they also take care of the colony here. One difference is that the ants were all adult ants who were taking care of the colony, but here the white termites are actually baby termites, whom in technical terms we call the nymphs. So the nymphs as well as the adult workers, they all take care of the colony here. Okay, and these colonies are made with vegetative substrate, but mainly soil and their fecal matter. So they feed on stuff and they defecate, and they use that to cement the soil together, and they make a gallery in which they cultivate these fungi. Okay. Now the termites, just like ants, they also have exactly same symbiotic association with fungi. In case of ants, we call it Atomyces fungi. Here we call it Termitomyces fungi. Okay, and here also in the colonies of termites, we find similar types of fungal parasites. But there we found Ascovopsis, and here we find another notorious parasitic fungus, which is known as Pseudoxylaria. Okay, so Pseudoxylaria, again, if you remove the termites, they will spoil the colony in no time. So I'm not going into the detail of this work because this is not I am not the main author of this paper. I'm just one of the contributing authors. So I'm just going to talk a little bit about this paper too. Here also we mimicked the study like we did in ants. And here also we found many different antibiotic secreting bacteria. Particularly there was one Pseudomonas strains there we had Pseudonocardia, and here we found Pseudomonas. Okay, Pseudomonas, Bacillus, Burkholderia, etc. There are many different strains of bacteria which also secrete antibiotics. Okay, and those antibiotics also have generalized antifungal activity. Okay, and Pseudomonas, this particular strain, this one was unique because this one killed Pseudoxylaria or stopped Pseudoxylaria, but it did not affect the cultivar fungus of termites. So there are some bacterial strains which the termites might be using a lot more because they do not damage the cultivars, but they damage only the parasitic fungus. But again, just like the ant system, here also we find many different antibiotic secreting bacteria which also can stop many different fungi, and they may not have effect on certain other fungi too. They may or may not kill Pseudoxylaria, but these are the particular bacteria which had at least some effect on Pseudoxylaria. Okay, so these are all the Pseudoxylaria control plates, and these are the Pseudoxylaria against the bacterial effect. Okay, so you can see that the bacteria is stopping the Pseudoxylaria. So this paper should come out within a few days. This is in press. If you are interested to know about our work, please go ahead and read it. It will come out in Microbial Ecology in a few days. Acknowledgement time. I have learned all this work from my postdoc supervisor, Professor Ulrich Müller from University of Texas at Austin. And these are my collaborators in the ant group, and these are my collaborators in the termite group. And for photos, I would like to thank Alex Wild. And most of the pictures which were uncredited, they were from pixabay.com. Okay, and I would like to thank you for your patience. And if you have any questions, I would love to answer them.
Okay, thank you, ma'am, for sharing such as interesting experience with us. Now I am requesting all of you to share your queries or questions, if any, below the chat box. Ma'am, there are some questions already. First one is, what black magic is this? This said from by Prachurya Ghosh. Prachurya Ghosh said, what black magic is this?
Ruchira, we can't hear you.
Okay, I'm just saying if you can switch on your mic and then ask the question directly, that would be better. Bapadita, you need to switch off your mic.
Oh, okay, ma'am. Okay, ma'am.
Yeah, so I'm going to switch off my mic. You can switch on your mic and directly ask the question, what do you mean by black magic? Are you still there, Prachurya? Okay, so anyway, this is no magic. This is just evolution. This is just innovation in non-human societies. Okay, so it should be taken as that. It is no black magic. There is no black magic, only science. Okay, any other question?
Bappadita, there are questions coming on.
Yes, ma'am. Next question is, ma'am, from Tulika Chakraborty. What is vortex? What is vortex?
All right. Vortex is basically a machine. Bapadita,
yes, ma'am.
So vortex is a small machine which basically will shake things very vigorously or very fast. You can increase the speed of that vortex and it just goes on moving like this. Okay. So when we had the ants in the tube, then with the saline buffer, we put them in vortex, and the vortex would shake it so fast that if there are bacteria on the body of the ants, then that bacteria would come out in the saline buffer, and then we isolated the bacteria from the saline buffer. Okay. All right. Let's go through.
Yes, ma'am. Next question is from Dr. Arunachal Chatterjee. What is the implication of studying agriculture in ants? What is the yes?
Sure. So agriculture in ants is basically a fantastic system to study symbiosis as well as the ecological importance of these ants. As I said, that these ants are pests, especially the leafcutter ants. So there are two types of fungus growing ants. Okay, fungus growing ants, all are fungus growing ants, but there are some which are more advanced, like Atta and Acromyrmex, and they actually cut leaves to make the substrate. Okay, so they are a little more advanced, little more complex. And as I said, that if you go in the forests of Panama or Brazil or any such other South American countries, then in these forests you would find long trails of leafcutter ants, and they are continuously cutting pieces of leaves. Okay, so they are very huge pests. Okay, so that is a problem. So you want to study that for that, and also this evolutionary system, this co-evolution model and all that. This is a plethora of interesting topics in this system. So that is the implication of studying that. Okay,
ma'am. The next from Atindra Kadar. Why there is less chance of fungus ant in India?
Say that again. Why
there is less chance of fungus ant in India?
Okay. So you see that the ants which cultivate fungus, they evolved in the New World, that is North and South America, and the termites evolved in Africa and India. There is the only problem of Atlantic. Okay, so Atlantic Ocean separates them out. So they have evolved and they cannot cross the barrier of Atlantic Ocean, and they cannot come here. Okay, so thank God they cannot come here, otherwise our forests will just go off. So, but that is an interesting system to study for scientists like us. So it is our misfortune that they are not there, but probably from the forestry point of view, it is a good thing.
Ma'am, last one, Prachurya Ghosh said again that since these antibiotic producing bacteria are found in the gut of the queen, and it is not the queen but the workers who are involved in cultivation, then how is the transmission from the workers to the queen achieved?
Okay, they are not found in the guts. I showed you a picture where there was below the prothorax. Prothorax is the first segment of the thorax, right? So below the prothorax, there is an area where these antibiotic secreting bacteria are found mainly, but we have reasons to believe that they are not only found in the prothorax, but they are also maybe found in other places on the body. Okay, so cuticle is the place, or the chitin basically on the insects. The exoskeleton of the insects is the one where the bacteria is growing. So for the workers to acquire that bacteria from the queen is pretty easy, right? So it is not coming from the gut. It is from the exoskeleton or the outer covering of the insects.
Then the last question is from Ayana, ma'am, why they choose many fungi strains together than just one?
We don't know. We don't know. We were actually expecting that they would take only one fungus, but they actually took all of it together. And we have done other experiments later on where we mixed the fungus and gave it to the ants, or we gave just two fungi and tried to see what the ants would do. And Attatoxina ants, they always mix the gardens. And if you actually just keep alive colonies of acromyrmex. I had done one experiment with acromyrmex colonies where I had put 3 different colonies in 3 boxes and connected them with those plastic tubes. There also I saw that the ants just went from one colony to the other. They collected all the fungus. They just brought them together. The most dominant colony would just go and take all the stuff from other colonies, and they would bring them together and they would make one garden. Okay, so we don't know why they do it. In nature, we find only one strain. But when we do these experiments in labs, then they just mix it up and they make it survive. So we don't know how they do it, but this is a very interesting phenomenon.
Yes, ma'am. Yes, ma'am.
Okay, ma'am. Thank you. Thanks a lot. Now, I am about to pass the microphone over to my departmental colleague, Mr. Moinak Mukherjee, for the second half of the... Yes, ma'am. Yes, ma'am.
There is one more question.
Okay.
Can you address that, please?
Yes, ma'am. Yes, ma'am. Sorry.
Thank you.
Oh. Yes, ma'am. Bodhisattva Chaudhary mentioned that you had mentioned that the ants produce chimeric gardens in the lab setting. Could you please explain the mechanism by which negative interactions between different genotypes of fungi is prevented?
Well, we don't know. Because in labs, if we put 2 fungi together in a plate, then the 2 fungi would kind of spray nasty stuff to each other, which we call the chemical warfare. But when it comes to ants, probably those burning areas, they just eat up. Or they have some mechanism to keep all these different genotypes surviving in pretty healthy shape. Okay, so I just worked 3 years on that project. So I tried many different experiments, but this particular thing, that how they are keeping it alive, this part would have taken much more time, and I did not have that much time. So I didn't do that. But the fact is that they mix it up, and how you can do is that you can select fungal tufts from different parts of these small gardens that the ants make, and then you try to sequence the fungi from different areas, and you see that these fungi are actually different genotypes. So they put them together. But how do they do it behaviorally? I can tell you, they just bring the parts and they put it together. But how they stop the fungi from fighting with each other, that mechanism, I don't know. Okay, we need to work more on that.
Ma'am, last one from Sarmishta Majhi. How ants balance between fungal growth and antifungus activities bacteria?
Very interesting question. So here, see, the fungus is going outside their body, right? So they are making a garden, and in that garden, they are raising that fungus. And the bacteria are growing on their cuticle. Now, cuticle, as you know, that this is kind of a dead material, right? So this does not grow, but the ants have certain ways of providing nutrients, particularly in the prothorax area, so that the bacteria, which require very little nutrients, can grow on these areas. So these bacteria are win-win things for the ants, because they take very little nutrients, they grow on their body, and they secrete antibiotics. Now, you should know, I'm sure that you know that on our body also, there are millions of different bacteria which are surviving on us. Some are pathogenic, but most of them are neutral. Okay, and our body can very well survive against those bacteria. But we do other things too. I'm very sure there are at least 5 billion bacteria on me right now, but I'm still giving a talk. So for ants also, it is just the same thing. They have some bacteria on them, but for them to cultivate fungus is not difficult. And when they interact with the garden, we expect that those secreting antibiotics are either actively put by the ants on the fungus wherever there is parasite, or it is a coincidence that the fungal strains come across with those antibiotic secreting bacteria and they die. Okay, so the parasitic fungi would die. See, I showed you a video as well as a picture of ants gardens where you must have seen that there are many bacteria in these, many ants in these gardens. Each of them have those bacterial secretions, right? So I think they can handle that pretty easily. They can use those antibiotics on the parasitic fungi, and when the parasitic fungi comes across that antibiotic secreting bacteria, they just die. Yeah.
Okay, ma'am. Thank you. Thanks a lot. Now I am about to pass the microphone over to my departmental colleague, Mr. Moinak Mukherjee, for the second half of the technical session. Moinak, please.
Thank you, Mr. Patro. It was indeed a very interesting session, and I am here to introduce our next speaker of the evening, Dr. ANINDITA BHADRA. It is my immense pleasure to introduce an alumnus of both my college and university. She did her graduation from Department of Zoology, Vivekananda College, Shakurpukur, before completing Masters in Zoology from University of Calcutta. Dr. Bhadra too was awarded a PhD in Behavioral Science under the guidance of Professor Raghavendra Gadakar from IISc Bangalore. She is currently working as an Associate Professor and Associate Dean in Indian Institute of Science, Education and Research, IISER, Kolkata. During her academic career, she has got numerous awards and recognitions and is member of many committees and editorial boards of several journals. Dr. Bhadra's current research focuses on behavior of free-ranging dogs. She has over 30 publications and book reviews, and several vernacular writings of her are available on the animal behavior topics. She has delivered over 100 lectures both in India and abroad. We are very glad to have her as our speaker for this session. Her talk on the great Indian joint families of free-ranging dogs will definitely give an insight to the world of free-ranging dogs. Now I pass on the microphone to Dr. ANINDITA BHADRA, madam. Madam, it's now over to you.
Thank you, Moina. I'll just wait till I can share my screen. Very good evening to everyone. Am I clearly audible?
Yes. Yes, you're audible and visible both.
Okay. So thank you very much for the kind introduction, Moina. I will begin by thanking the organizers of today's session. And it's a great pleasure to be here listening to my friend from master's years, PhD years, lab mate, colleague, long-term friend, Ruchira. It was a great pleasure listening to her, and it's an extra privilege that both of us are here. We've done our PhDs and masters together, and thanks to Changjukta, who has brought us here together on the same platform today, and to Pukichat College. I would like to begin by thanking the very illustrious organizers who have thought of doing this virtual symposium, and special thanks to the principal, Dr. Chondo, and the IQAC coordinator, Dr. Pal, for their kind introductory words. So without much ado, let me move on with what I want to talk about. Before I tell you about my work or our work on free-ranging dogs, let me just give you a glimpse of where I come from. This is the IISER Kolkata campus. We have a 201-acre campus away from the den of the city, though we are called IISER Kolkata. I think most of you would know that we are not in the city. We are in a place called Mohanpur, which is somewhere in between Kachrapada and Colonies, and you can reach us within an hour to 2 hours, depending on where in Kolkata you start from. And you can come to this very green and beautiful campus, which also boasts of a fantastic auditorium and a fantastic swimming pool, which our students enjoy before the pandemic, which is of course unfortunately closed now. And we invite people to visit us. We always welcome teams of students and teachers who would like to come for an educational day trip and visit our labs in different departments. It's a very simple process. You write to me, the associate dean, and I organize this. And we are, after the pandemic, when things open up, we are actually going to try and set up an online application process to streamline this. So you are most welcome to visit us whenever nature permits. So IISER Kolkata is, this is our hostel complex, and we are very proud of our flagship program. All the IISERs, for that matter, have this integrated BSMS 5-year program that make us very proud. We have excellent students joining us, but we also have an integrated PhD or post-BSc integrated PhD program and a PhD program, which where we get students like you who join us after their graduation or masters. And we encourage you to apply, come visit us, try to be here with us for your PhDs if you would like. As you can see in this map below, this is not a very well updated one, but this is a map which had been created by our alumni association, showing the footprint, academic footprint of our students all over the world. And we are very proud that some of our early generation students have now become faculty in important institutes in India, like the IITs. We have a very interdisciplinary academic ambience where we encourage students to work across departments, across topics, and that is one of the signatures of the IISERs, the 7 IISERs across India. Our lab is called the Dog Lab. Very obviously, we work on dogs, and this is part of a group of people who work on behavior and ecology. There are actually 3 alumni who work on behavior, all of us from the Center for Ecological Sciences, so it's a very nice place to be with old friends. And we particularly study behavior, ecology, and now cognition of street dogs, stray dogs, free-ranging dogs, as you would like to call them, in different parts of India. And of course, majority of work happens in and around our campus. When I tell people that I work on dogs, they often ask me, oh, which breed? Because even a couple of hours back, I was talking to somebody on the phone about, whether street dogs are a different species from the different breeds of dog that we know of. And I was trying to tell him, no, they're not. And people often ask, oh, are the street dogs the same as wild dogs? No, they are not. Wild dogs are wolves. The street dogs that we look around, we see around us, are dogs as much as any of these breeds. All of these, the breeds that we know across the world, are products of artificial selection, much like all the crops that we grow, most of the fruits that we eat today are products of artificial selection. We have selected out characteristics of individuals, and we have practiced breeding to get animals which we thought were better. We have Jersey cows, we have the horses, which are really big stallions. None of these are products of natural selection. They're products of artificial selection. So are these breed dogs. And I have to tell people, no, sorry, I don't work on breed dogs. I work on free-ranging dogs. Pet dogs, in whatever form you get them, they are exposed to a lot of care by humans. So we enable them to mate, we enable their birthing process. Some dogs are by default born out of cesarean sections. We hand rear them, we give them a lot of luxury and comfort, and we vaccinate them, and we ensure their survival. Whereas dogs on streets have only their mothers and their relatives around them, and they have to go through all the quote-unquote struggle for survival. And a lot of our work focuses on understanding how they survive on the streets. Interestingly, when people talk about the evolution of dogs, we are very fascinated by the dog because we call them man's best friend. We know that dogs and humans share a special bond. And a lot of people across the world are trying to understand how this special bond developed, because we know this is the product or end product of the domestication process that dogs have undergone. Interestingly, all other domestic animals have undergone domestication, but very few of them live with us in their homes like the dogs do. You know, some people might say, oh yes, I take my pet cat to bed, or I take my pet rabbit to bed. But if you think about it, cows and horses and pigs have been domesticated for a long, long time. But then you use them for certain purposes. They are domesticated animals. They are taken care of, but you don't spend your days with them. You don't go take them on long walks. You don't take them to your bed. You don't take them to your dining room like you do with your dogs. And this is why dogs and humans share a special bond, and a lot of scientists try to understand how this evolved. We know that wolves or wolf-like animals are the ancestors of dogs. In fact, to students of zoology, you would also know that wolves, even today, they are Canis lupus lupus, whereas dogs are Canis lupus familiaris. So dogs and wolves are still capable of interbreeding and producing viable offspring. But then we also know that a branch of wolves or wolf-like animals underwent domestication, became dogs. And the question is, what happened during this process that made them man's best friend? And people try to study pet dogs and compare them with semi-captive or captive wolves to understand this process. What we don't know most of the time, though we think of dogs as the pets, majority, as high as 70 to 80% of the total dog population of the world exists as free-ranging dogs. They are not really the pets that we see around us. And also, if you think about the evolutionary perspective, then the free-ranging condition is closer to the ancestral condition in which dogs lived around humans, probably as commensals and scavenging on leftovers from humans. Eventually building a relationship with the people is the kind of condition in which they can be studied best if you look at the dogs in the streets. And of course, that through that process, we can understand the process of dog domestication, at least to some extent. If you look around the world, then all the red marked countries here are actually places where you can find free-ranging dogs. And one place that is missing in the north is Russia, where you can also find free-ranging dogs in some cities, especially in Moscow. So if you see, it's primarily the countries which belong to the global south where you find free-ranging dog populations. Countries in Africa, Southeast Asia, South America, and of course, in the Indian subcontinent, you find free-ranging dogs. And still, the notion of the dog, of what the dog should be and where the dog should live and how it should be treated, comes to us from the Western world or the global north. If you look at the Indian situation, of course, anybody living in India has always encountered free-ranging dogs all their lives. And no matter wherever you go, if you have traveled across the country, you know from the mountaintops to the seasides, you are going to come across free-ranging dogs. And if you are one of those people who like dogs, you must have befriended dogs in all kinds of places, wherever you've gone. Dogs share a love-hate relationship with humans. And this is one of those, happy stories which I always try to talk about. This is a new story from 3 years back or 2.5 years back, which I happened to come across. And this made me very happy because this was about this one dog which had followed a group of mountaineers up a very high and tedious peak in the Himalayas. And this was definitely not dog habitat, but this dog just happened to follow this team. And at the summit, they decided to share some of their food with the dog and show it some love. This reminded me of another story which every Indian probably knows of. If I show you this picture, I'm sure you know what story I'm talking about. Any guesses?
Mahabharata.
Yes, this is Mahabharata. the ending
মহাপ্রস্থান
where
পথে
yes,
মহাপ্রস্থানের পথে
right, Mahaprasthaner Path. So this is where you have the 5 Pandavas, and then there's a dog, and here is Draupadi who has already fallen down, and we know what's going to happen. These dogs are this dog is going to follow Yudhishthir up to heaven, and eventually we'll get to know that this is Dharma disguised as a dog. But why I like the story is because it tells me something else about dogs and humans, even when the Mahabharata was being written down, and probably even earlier when the Mahabharata was not even a written text but a story being told by people in the Shruti culture, which is dogs lived around humans, followed around people even then, as they do now. This dog making it to the summit with Yudhishthir is no different from the dog in the new story making it to the summit with the mountaineers in 2018. It is not about whether this was dharma or not, but the fact that the behavior has not really changed much. This is a dog that we can all relate to in our day-to-day lives. Especially if you are a person who likes dogs, then I'm sure you have been followed by a dog out of its territory and comfort zone into a foreign territory. Just because you were going, the dog followed you. This is an experience that many of us have shared, and Ruchira would be very aware of a famous dog called Chakli, who used to do this every day, running behind my cycle and spending the day in the lab while I was there and going back home with me. And everybody in the department came to visit her and feed her biscuits. This was a random stray dog who came and adopted us one fine day. And this is a very, very natural behavior of our stray dogs. Chakli even used to attend our lab meetings, by the way. So when we study animal behavior, people like us, we try to put our questions in a broad framework, and classically there are 4 kinds of questions you can ask. One is a question about the mechanism or how a behavior occurs in an individual, the ontogeny or the development of the behavior, how it arises in an individual or how it changes from a juvenile to adult condition, the function or adaptive value of the behavior. Why is this behavior there in the species? What adaptive value does it have? And the phylogeny or evolution of the behavior, how this behavior arose in the species and not in the individual. And on the right side of my slide, I remember here the great Nicotin Virgin, who put down this broad framework. And these are called Tinbergen's 4 questions, classically known as Tinbergen's 4 questions. And Nicotin Virgin was one of the 3 people who together shared a Nobel Prize in medicine for studying animal behavior once until now in history. The other 2 were, of course, Konrad Lorenz and Carl von Frisch. So we divide these questions into proximate questions or how questions and ultimate questions or why questions. So we broadly try to answer the question of how an animal lives and why an animal lives the way it does. Those are the how and why questions in evolution, which we try to address through our studies in animal behavior. And I have tried to put some of these questions down into our work on dog behavior. So in the dog lab, we try to understand how dogs interact with each other within social groups and between social groups. So we try to look at behaviors like mating, parental care, interactions between siblings when they are juveniles or when they're adults, interactions between individuals for maintenance of territories, which is something that we are all aware of. Who has not hated dogs for barking in the middle of the night just when you're trying to fall asleep, or when you are watching a movie on Netflix and dogs start howling just outside your window, or you're giving a talk and dogs start howling outside your window? You don't really love them that much. And we also try to look at competition over food, space, and mates. We try to understand how dogs interact with humans, and this this is an aspect of our work where we can put our work in the framework of much bigger body of literature that exists from other labs who have studied dogs, because most people who work on dog behavior work on pet dogs and try to look at dog-human interactions. And in this framework, we can ask our own questions on dogs and human interactions. We try to understand how humans can act as resources for the dogs, how humans act as threats for the dogs. And we also try to understand how quote-unquote smart dogs are, in the sense of their ability to solve tasks. We have worked typically on problem-solving ability in terms of physical, cognitive tasks like opening a container and getting food, or finding out food from a box containing a lot of garbage in which food is hidden, to basically try and understand scavenging behavior. We have tried to understand their ability to distinguish between quantities. Can they distinguish between less and more resources and choose one from the other? We are looking extensively at food preference and how food preference is applied in their natural behavior of scavenging. And in doing so, we are trying to piece together tiny pieces of the big picture in asking, how do dogs become man's best friend? And at the end of 12 years of work, we have got little bits of answers, but of course, there's much more work to be done. And the interesting part is, you address one question, you find an answer, it opens up 5 new questions, and that's how science progresses. So today I'm going to give you kind of glimpses into some of the work we have done, primarily based on behavior, and that's why the title of the Great Indian Joint Families. And at the end of this section, I hope I'll convince you that dogs do live in joint families like we do in India. Some of us do. So let's take a look at the first few months of a dog's life. We know that we have parental care in all mammals, at least in the sense that mothers have to nurse their offspring, otherwise the offspring will not survive, that they are mammals. And I'm sure all of you are aware that when puppies are born, their eyes are not open, they're very immature, they cannot move, they cannot help themselves, and the mother has to take a lot of care. So in a series of studies, we have found a lot of data which is put together in this one infographic. It's a social network where all the light blue circles are pups from one litter, and this dark blue circle represents pups from a non-sib litter, so pups of another female. And if you see here, the yellow circle is a human interaction, so all human interactions have been put together in the yellow circle. The red circle is the mother. And just hold on for some time and I'll explain to you what the putative father and the allo mother are. But what I wanted to show you is, as soon as a pup is born and as it is growing, this poor pup is actually facing a lot of complex social interactions. And this is only from the perspective of play behavior, because play is something that pups do very often, and as they are growing, they are playing a lot. So this is one social network which I have just shown you to tell you how complex a pup's social life can be. And to this, if I added all the interactions that a pup initiates or receives from other members of the group, then you would not be able to even make out the circles. So this is just play, and this is as complex as it can go. We did a 5-year-long long study to understand whether mothers actually try to find suitable dens before they give birth, and what kind of spaces they adopt as dens. Because we thought that if the mother is putting in some effort to select a den, then her care for her unborn offspring start with that process, because she has to put in effort, spend energy and time in locating a den and giving birth there. So that is also a form of parental care, but pre-birth. And we found dens of all kinds. You can see here, this is a garbage dump. This is somebody's backyard with a lot of garbage strewn around. This is actually on our old campus where somebody had given them some rugs to lie on. This was interesting because this is one of my favorites. Somebody's old abandoned sofa set in which this female had given birth. Here, this is a place to store stuff in somebody's backyard where she has gone and given birth. And here you see they have actually made a nice den for her because this dog is associated with the household here. So all kinds of spaces, and we actually collected data on a large number of these dens, 105 to be precise, and we did measurements of the space available. We took data on whether it's open or covered, so how much shelter it provides, how far it is from the nearest watering site, how far is it from the nearest source of food, how far is it from human disturbance, and we put all of this down, qualitative information down, and converted this into a scaled matrix. And we found that, I'm sorry, I think I have, okay, I missed out this image, but let me just tell you, we found that the mothers are actually not shying away from human presence. They are actually preferring to den close to human settlements, as you see in these images, and they are actually preferring human given food rather than going out and scavenging by themselves, leaving the pups alone for a long time. So this was the first thing that we noted, that all urban animals that have been studied so far are known to avoid humans while they are giving birth. Their dens and nests are typically away from humans or hidden, or they are very protective about it. But for dogs, it is the other way around. They are actually seeking out human proximity, depending more on food, more on humans for food, and trying to be close to humans as if the humans are a resource. And then when we started looking at parental care behavior in the pups, you note here that our data starts at age 3 weeks. So the first 2 weeks, we don't have data, simply because typically the mothers are very protective. They curl up around the pups, don't let us go close enough to observe them, and we don't want to disturb them to an extent where the mothers will run away with the pups and we lose them. So we don't disturb them for the first 2 weeks when the mothers are ultra protective. We just go there, sit around, get habituated to them, let them get habituated to us, and 3 weeks onwards, there is behavioral observation done on the mother and pup unit. And this graph here gives you an overview of the types of behaviors that were seen. This is called a time activity budget, which basically means we are seeing different kinds of behaviors, and the dog's time that we are observing has been spent in these different behaviors. Now you convert this into proportion of time spent by the mother in the various behaviors like nursing or suckling, like allo grooming, which is cleaning up our pups, licking them, removing insects from their bodies, piling up around them and giving them protection, playing with them, giving them regurgitated food, which is something which starts a little around 5 weeks and continues for some time. Protection, which is their barking at others, could be people, could be other dogs, chewing them away from where their pups are. Sometimes the mothers would get solid food, bring them in their mouth and drop it in front of the pups and let them eat. So that's offering of food. And they will clean up the area by eating the fecal matter of the pups when the pups are very small. So all of these behaviors, we put this in this chart where each bar is broken up into the way the behaviors are distributed. And you can see that as the pups are growing, the time spent in each of these behaviors is changing. The most notable is the behavior of suckling or nursing. You see here, initially there's a lot more time spent in suckling, and that almost goes down to 0. 14 weeks onwards, there is no suckling. Play increases as the pups are older, as does protective behavior, whereas regurgitation, which is the pink, is hardly visible here. You see it here. So different ages of pups, the mother is spending different amounts of time in each behavior. And thus, we know that the mother is adjusting her parental care levels in both quality and quantity. We tried to look into this in a little more detail. Don't get worried about the complexity of the graph. We have 3 parameters here. So this is a 3D graph. Here, the age of the pups increases from right to left. Follow the arrow. The litter size increases from up to down. And the proportion of time which is spent in active care increases from bottom to up. So here you have the smallest litter sizes at the earliest ages. So the smallest pups in the smallest group which receive the maximum care. And the minimum care is at the valley of this, where you have the smaller groups with, even at a smaller age, for a higher litter size, you have much less care. And for higher age and smaller litter size, again, you have much less care. So basically, you have sibling competition, and as the group size of the litter increases, the care received per pup decreases. So the mothers can regulate the level of active care they give to the pups to a certain extent, but the mother's ability to give care is limited by her energy levels, right? And her time. She cannot give infinite levels of care. So what happens is, if a mother gives birth to 3 pups, 4 pups, 5 pups, the pups can get an optimum level of care. But if a mother has a litter of 10 pups, the amount of care that each of the pups will receive is very low, and the chances of survival of an individual pup will go down. So for pups, it is advantageous to have less siblings, but maybe not 0 siblings, because siblings are also going to provide social interactions. Now we looked at nursing behavior in a little more detail, because that is definitely the most important and most energy intensive kind of mothering that a female would give to her offspring. And we found that though the mothers are continuing to give care when the pups are 14 weeks, 15 weeks, 16 weeks old, nursing has pretty much stopped after the 13th week. So there is a time after which the mother is not nursing anymore. Not only that, from the 3rd week to the 13th week, there is a constant decay at the level at which nursing happens. And we were interested in understanding the process of weaning, because weaning has a very interesting evolutionary implication. Before that, let me try and play this video for you. There's no sound, so don't worry about the sound. And these are scenes which I'm sure all of you have seen at some point of time or the other. Often what we have seen is people would grab the mother and make her stand until the pups are able to suckle from her. And the general notion is, oh, look at this mother, she's being so nasty. What kind of mother is she? She is not, agreeing to let her offspring suckle from her. And that is the human notion that we add to the behavior of the mother. But when we looked at the data, we found, pay attention, there are these red triangles, which is the proportion of suckling bouts or nursing bouts, or whatever we would like to call it. Typically, from the perspective of the pup, it is called suckling. From the perspective of the mother, it is called nursing. So the same behavior. But when it is initiated by pups, we mark them by the red triangles. When it is initiated by the mothers, we mark them by the open circles. If you look at this data, notice that in the 3rd week, 4th week, 5th week, all the bouts between the red and the red triangles and the open circles are kind of distributed in 50-50 proportions. 6th week onwards, the mother-initiated bouts are lower, whereas the pup-initiated bouts are above 50%. Then you see here, the open circles reduce. 9th week, 10th week, there are no mother-initiated bouts after the 10th week. That is when mother has stopped initiating nursing. She's not interested in nursing anymore. And all the nursing you see is like these videos where the pups try to grab her and suckle from her as much as they can until she runs away. And this is when weaning has started. This we found to be very interesting because this window of weaning is something that uh, immediately helps us to test a famous theory in evolution known as parent-offspring conflict theory, which was given way back in 1974 by Robert Trivers. Don't worry about the complicated graph. What Trivers basically was trying to explain was why weaning happens. In principle, we should think that mothers should be always ready to give care to their offspring, right? That is what a mother's behavior is expected to be, at least in the human sense. And he was giving this theory from the perspective of human behavior, not any other animals. He was saying, even in humans, mothers go through a process of weaning their offspring from lactation or from nursing. Why do they do this? And he gave this based on genetics. He said, for every mother, each offspring carries 50% of her genes. And a mother would want to maximize her reproductive fitness by producing more and more offspring. Now her energy is limited. So once she has nursed an offspring, ensure that this offspring is big enough to not survive and eat other kinds of food, mother should wean this offspring, replenish her reserves, and invest in the next offspring. So she should pay attention to her lifetime reproductive success. Now, from the perspective of the offspring, I am related to myself by 100% relatedness to me. I am related to my siblings by 0.5 or 50%. I share 50% of my genes with my sister or brother. So, from the perspective of the offspring should also want the mother to give birth to more siblings because that gives more inclusive fitness. But then it should love itself more. So, there will be this window when initially both mother and offspring should agree. When I say agree, I don't mean they sit across the table and have a meeting and discuss and agree, but biologically agree that parental care is necessary and the mother should give care. After this window, the offspring and mother should agree that care is no longer required for this offspring. Mother should invest in the new offspring. But within this window, there will be a conflict where the mother would want to wean and the offspring would want to get a little more care out of the mother, and this will give rise to the weaning conflict. And this data gave us a beautiful demonstration of weaning conflict happening. And we looked at this, initiation of the behavior. Just by looking at the rate of nursing, we were not seeing this. But when we divided it up into who initiates the behavior, we immediately saw this difference. We also looked at it in a little more detail. And the next step that we found was the duration of each bout of nursing. So my student Manavi, who was doing this work, every time a nursing bout started, she would start her stopwatch. Every time it ended, she would stop her stopwatch. By doing this, she could differentiate the durations of the mother-initiated and the pup-initiated bouts of nursing and suckling. The red ones are the mother-initiated bouts, the blue ones are the pup-initiated bouts. And you see here, 8 onwards, there are no mother-initiated bouts, so their duration is 0. So this matched very nicely with the initiation behavior data. And the two windows were the same. So we identified the 8th to 13th week window in the pup's age as the weaning period in the dogs. So Manavi created a timeline for development of the pups, and she said that up to 7 weeks, there is high level of active maternal care. Beyond 13 weeks, there is mostly passive maternal care. And in between, there is a period of parent-offspring conflict. Beyond 16 weeks, there is no maternal care at all. And between 13 weeks to 16 weeks, we call the pups as juveniles when they are not actively getting maternal care anymore. And then they become subadults and adults. Now, we said, let us pay a little more attention to this weaning conflict and try to understand how selfish mothers can be. Can we make the mothers act selfishly? And we realized that one thing that is happening from our behavioral observations, we noted when the pups are small, people become very tolerant of the mother dogs. They give them extra food. They say, oh, poor mother, see, she is losing so much weight. And they give a lot of food. When the puppies grow a little older, they become a menace and people don't feed them anymore as much as they were doing earlier. So this is a window of time when the mother can actually grab more food or she can get more food to feed to her offspring. So we said, let us go test parent-offspring conflict theory by doing an experiment. A simple experiment because those were the early days of my lab. We didn't have much resources. All we needed was a video camera, a notebook, and some biscuits. So we asked whether there is parent-offspring conflict over extended resources in dogs, which is extended parental care, which is in this case human-given food. And the second question we asked is, would the quality of resource affect the cooperation between mother and her offspring? So I'll first show you what we did with the first experiment. So in the two videos here, they're playing simultaneously. If you notice, there would be food given. The video is running a bit slowly, but I think you can make out. The first one, the mother is not trying to grab the food. That is the bottom line of it. The mother allows the offspring to take the food. The second, very aggressive. Here. So she is actually trying to bite the pup and stopping it from getting to the food. We did experiments of this kind with 15 mothers and their offspring for a continuous time from 8th week of age of each litter to the 14th week of age. And we found very nice data where the proportion of conflict increased steadily as the pups grew older. And this was really nice because we said, yes, as the pups are growing older, mother is competing more and more with her offspring to grab food. Not only that, we actually tried to measure the body condition of the mother. So all we did is from the videos, we scored the mother on the first day of the experiment, the last day of the experiment, and the midpoint of the experiment for scars on her body, whether her ribs are showing, whether her skin is looking shaggy, whether she has become very thin, all of this together. And again, this qualitative data we converted into a graded score and we put them together. And I think it's quite evident from just the graphs that between the green bars and the dark brown bars, there is a significant difference. And when we ran this data through statistical analysis, we found that yes, for all the mothers, there was a significant improvement in her body condition from the first day to the last day of the experiment between mothers at the same stage. So all mothers on the first day of the experiment, when all their pups are 8 weeks old, all mothers when their pups are 14 weeks old, between the mothers, there isn't significant variation. So among the green bars, among the brown bars, it is not different. Whereas from the green to the brown, there is a significant positive change in the mother's body condition. So the conflict over the food was not getting converted into milk for their offspring, but getting converted into energy for the mothers to replenish her reserves. That was the point of it. Then we said, okay, we got help from a friend in physics to model what would an altruistic mother look like and what would a selfish mother look like if we started from this kind of a data. So in both the graphs, line 1 denotes the experiment with the biscuits. Line 2 is an hypothetical line of if the mother had this selfish strategy, that is, she showed more was altruistic, that is, she wanted to share more because richer resources were available. I need the very rich resource and the mother's offspring if she were to share this with the offspring. So we had selfish strategies and we had altruistic strategies. And then Manavi went and did the same experiment with another fresh set of mothers and their offspring in the next year. Again, the video is taking its own sweet time to play. I'm sorry about that. So here you will see all the different behaviors in one piece of video. I will keep my video off so that the bandwidth improves. It's getting very slow. This one is my favorite. The puppy actually tries to grab the food out of the mother's mouth in this case. Okay. Finally, when we did the experiment, the videos are just an example, but with the data, you see here, this is the graph on the right side is the theoretical graph, and the graph, the bigger one, is our data. And this was such a close match that initially I was very worried that we have made some mistake, because this is too good to be true. But it turned out that this was really the data, and the mother was indeed showing the selfish strategy. And this matched excellently with the predictive model that had been worked out. And we said, yes, the mothers behave more selfishly when you give them a richer source of nutrition. So if I were to sum this up, with their pups, during the weaning phase, the slides are taking a bit other. I'll just wait until I will go to the next slide. I've already, yeah. So the mothers are competing with the pups during weaning for extra food, which is coming from humans. And not only this, when you give them better food or better maximize their lifetime reproductive success rather than focusing on the current litter alone. So the pups more concerned about the survival of her lifetime offspring, all the offspring she can produce in a lifetime, rather than focusing on the current litter. And this was the first time an experimental field demonstration of POC had been done in a natural system. So I move on to the other part of the members of the joint family, which I call them helping aunts and then stealing pups. So the photograph here is actually of one mother with two sets of pups, one belonging to her and other belonging to another female in the group. We found a lot of groups in which there were two females, sometimes three, rarely three, but mostly two females who had given birth at the same time. So there was an overlap between their births. And this data comes from a large number of field-based observations on actually 23 litters or 23 mothers, but this multiple mother groups came from 15 dog groups where we had 19 such units. And if you look at the Venn diagram, the pink outer circle gives us groups, 5 groups in which we had only maternal care received by the puppies. Then you have the yellow circle here where there are 3 groups which, in addition to care by their own mothers, received care from some female in the group who's not the mother, but who's a lactating mother at the same time. 3 groups in this light green received maternal care plus care from some male in the group, which we called male allocare. Care our fathers because we don't have the genetic data. Dogs are promiscuously breeding animals. We know for all of these cases, the mothers have mated with these males, but we don't know whether the males are the real fathers. And there are 10 groups in which the pups have received care from all three: mothers, females other than mothers, and males, whom we call putative fathers. And outside all of this is this one group, which is a very special group because the day after the pups were born, the mother died in a car accident. And the grandmother, who was in the same group as the mother and was heavily pregnant, kind of adopted these pups. This happened the previous evening. The next morning, we go and see the grandmother is no longer pregnant and is taking care of her grandpups. So what we assumed happened, we did not know this, is probably she had a miscarriage because the pup started suckling from her. We spoke to a vet who said that this is possible during late pregnancy. If nursing starts, then the hormones will go berserk and there will be a miscarriage. So probably the grandmother gave birth to stillborn puppies, and when they do that, they eat them up, or jackals can come and eat them up. So we did not see those puppies. The grandmother had adopted her grandpups and was behaving like a mother. So we had maternal grandmothers, we had older sisters, and we had maternal aunts, so mother sisters, who have all been shown to give care. And that is why I call them the great Indian giant families of free-ranging dogs, because these are the Mausies, older Didis, and the Dadis and Nannies who are giving care to the puppies. Of course, when you have allocare, there is benefits due to kin selection to the females if they're all related, and there is benefit for the pups in their survival. We tried to look at the kind of care. We found that allocare mothers do provide care, but at a lower level from that of the mothers. Overall, the level of allocare given by the allo mothers is much lower than the level of care given by their mothers, which is to be expected. When we looked at the allo mother, the mother, and the putative father, we found that the putative father spends as much time caring for the pups as does the mother, which was very interesting. So we tried to look at what kinds of caring behaviors putative fathers can show. And I again have a video which I'll not play because it's again going to get stuck, but these were some of the behaviors that the putative fathers were showing, which is typically play or regurgitated food. But this is the graph that I had shown you for the mother. We created a similar graph for the putative fathers or the males who were showing care to the pups. Of course, the males could not show nursing behavior. That's not possible. But other than nursing, they showed all the other kinds of behaviors. When we did a comparative analysis of the different kinds of behaviors shown by the males and the mothers, we basically concluded while mothers are mostly using their energies to feed, the fathers are primarily showing play and protection behaviors. And we like to say it in a kind of sum it up in a fun kind of manner to say mothers feed, fathers play. We looked at another behavior because we thought it was very odd that mothers were also showing nursing. It made sense that they were protecting, grooming, playing, all of that made sense. But it didn't make sense to us to see that allo mothers were also nursing the allo pups. When they already had their own pups, why would they allow these pups to nurse from them? Now, the same data which we had initially showed of suckling, to that you now have allo mother initiated nursing bouts. And where do you see the purple dots? Nowhere here. All the purple dots are actually sitting on the x-axis, so you don't see them anywhere in the graph. Basically, what we found is out of 594 allosuckling bouts over 11 litters, 100% of the bouts were pup initiated. So the allo mother, we saw them allo nursing, but they were not doing it voluntarily. The pups don't care who they nurse from. They find any lactating female around them and go latch onto them and try to grab some milk. This is called milk theft. This is a behavior which is well known in ungulates, which live in large herds and move in large herds, where a lot of offspring are born to a lot of females at the same time, but has never been reported in carnivorous societies. And this was very amazing because we realized that the pups are basically in conflict with the allo mother. So what looked like all hunky-dory, nice aunts and grandmothers taking care of their pup, little pup relatives, this was not really so. And this data again showed the proportion of the bouts of each of these. So if you look at the allo mother mediated termination bouts, then all of them, 100% of them, are basically terminated by allo mothers. So they're initiated by the pups, allo mothers realize these are not mine, and they kick them off and run away. So that is what happens when allo mothers are nursing. So we concluded that there is really no allo nursing happening, which is all very nice and cooperative altruistic behavior, but the allo mothers or the females are basically victims of milk theft. Now, all of this put together, the pups are receiving a lot of care. So we wanted to look at pup survival. And this is the data of survivorship, which as you see, as the pups are growing older, age goes from 0 to 7 months. So each litter was followed up to 7 months of age or until they all died. And you see survivorship decreases as the pups grow older. And the maximum mortality happens when the pups are 4 months old. Remember, 4 months is approximately 16 weeks, which is when they have weaned, they have become independent of the mothers, become juveniles, and now they're roaming around on their own, and they're becoming victims of human cruelty and accidents. So pup mortality increased with age, and highest mortality of 34% in a month was observed at the 4th month. Maximum survival occurred at the first month of age, which is when they're completely under the care of the mother. We looked at the various causes of death. We looked at human influence death, which is when people poison them, beat them, and injure them to such an extent that they die. Then there are pups which are picked up from the litter often by kids, taken home and then abandoned, or natural deaths, and then of course unknown. And we saw that the majority of deaths are human influenced. So actually, when we summed it up, when we summed up, we actually found that only 19% of the pups born in a season survive until 7 months of age, which is the onset of adulthood for dogs. And of course, there will be further mortality. So when you see a lot of pups born in a season and people keep saying, oh, the population is increasing, population is not really increasing that fast. If 5 puppies are born, only 1 has a chance of survival. So it's not really as bad as we think it is. Putting all of this together, we drew up a very complicated timeline of a pup's life. We have phases of development where we have lowest mortality, starting of foraging, parent-offspring conflict, marking of highest mortality at fourth month. We have phases in which we have a lot of care. We have phases in which they show certain behaviors. And that is the summary that I would like to leave this part of the talk with before I conclude. So if I look at what I've been talking about, a mother dog shows extensive care towards her pups, starting from before they are born, when they are selecting dens about a month before the pup is born. Care is limited because when the litters grow larger, per unit of care decreases. So there is sibling competition from birth. Weaning conflict is very real. Mothers begin to compete with their pups for food during the weaning phase. Lactating females are often victims of milk theft, which adds to the survival of the female who's trying to wean and who's trying to replenish her reserves for lifetime reproductive success. So it's a complex, dynamic interaction. We have mother-pup interactions revealing cooperation conflict dynamics. We have helping relatives, we have stealing puppies, and we have the males who are probably the fathers, we don't know, which can enhance the survival of the pups. They are the first case among canids which present a communal breeding system. Typically in canid species, we have a cooperative breeding system, not a communal breeding system. All of this was part of my first student Manavi's thesis, and I was very proud and very happy when Professor Gadekar wrote an entire chapter in his series in Resonance, where he summed up all of this in the context of parent-offspring conflict. Not because this is part of this book, but because this is a fantastic book for every student. I urge you to go download this book, which is freely available on the internet, where he has spoken about 16 different experiments in animal behavior. Each chapter is a series of experiments, which are behavioral experiments, which can be done with a little thought, with a little bit of intelligence, and almost no cost. A fantastic book published by the Indian Academy of Science, written for students like you, and highly recommended. I will, just before I end, I just want to tell you that we also work on how we interact with humans. We do a lot of experiments on the streets, trying to understand how dogs adjust to humans, how well they understand human social gestures like pointing, like a threatening posture, how they make friends with humans, and how we humans influence their behavior. For example, recently there's a paper which has been accepted today, in which we try to understand how during Durga Puja, when we flood the roads and throng out in large numbers and disrupt the habitat and completely change it, how does that affect dog behavior? So this is ongoing work. So I'll just sum up that when it comes to dogs and humans, we have found that dogs are capable of understanding and utilizing various human gestures like pointing. And this was thought to be something which can only be happening in pet dogs, but now we have shown that this is ranging dogs can interact this way with complete strangers. We have found that adult dogs show a lot of plasticity in their trust on humans, and trust is built not because of food given to dogs, but they build trust on humans based on who shows them love and care, not based on who feeds them, which is, by the way, a very, very good survival strategy. And that agrees very well to the current understanding of even human evolution. I recommend this book, Survival of the Friendliest, which talks of how becoming friendly towards each other has helped human cultural evolution and has made us humans in the first place. This is a recent book, and our work on dogs is actually agreeing to a large extent what Brian Hare and Vanessa Woods talk about in terms of what makes us human. So friendliness might be an answer to the evolution of the dog-human relationship mystery. That is what we think at present. I would like to end by thanking the entire set of students who have contributed to the dog lab, and there are a large number of interns who are always there in the lab who contribute to our ongoing projects. And thank you for listening, and thanks to DST, SERB, CSIR, INSA for all the funding that I have received for the work, and of course for IISER Kolkata for all the support. And I'm happy to take any questions. Thank you.
Thank you, ma'am, for your very good and enriching lecture. Here are lots of questions. So I would like to read them to you.
Yes, sure.
The question is from Dr. Arunachal Chatterjee. The question is, what are meant by allo mother, allo care, and so on?
Allo care is care given to an offspring by someone other than the biological parent. So an allo mother is somebody who's showing a female who is showing care but is not the mother. Allo father would be a male who is showing parental care but is not the biological father.
Okay, the next question is from Jyoti Mehta. How do you keep track of so many pups?
With a lot of hard work and patience. So when I show you data from the behavioral work, it is based on data from individual groups. And to collect data on 23 groups, Manubi worked for 5 years. So this is 5 years data put together in one graph to show you. So each season, we can only observe a few litters because all of them are born at the same time. And for each group, you want to observe them from their third week to their